Yeah, welcome to my talk about marine macrophytes as carbon sinks, a comparison between two endemic plants and the invasive algae Halimeda incrassata. What I'm going to talk about is a short introduction, my aims, my study site, followed by my field study, then my laboratory experiments, a conclusion, and a prospect. For the introduction, since the 18th century, we humans, basically after the Industrial Revolution, increased the anthropogenic emissions of greenhouse gases, mainly CO2, constantly, which resulted in the latest Keeling curve update of an all-time high for 2019 of over 400 ppm in the atmosphere. And the IPCC, the International Panel of Climate Change, gave out predictions towards our future, like for the end of this century. In my study, I focused on the RCP 8.5, which basically states that it will be steadily keep on increasing till the end of this century, and the RCP 4.5, which will level off around or after 2070. The increased emissions has two consequences, which is water temperature rise and ocean acidification. Globally, 10 to 15% of carbon is buried underneath seagrass meadows, and the Mediterranean Sea especially is a hotspot for climate change. The seagrass meadows, which is the dominant coastal ecosystem in the Mediterranean, is especially prone to climate change, with additional other effects like invasive species, which ultimately leads to a tropicalization of the Mediterranean Sea. My aims of this study were to investigate the effects of global warming, ocean acidification, and the combination of both on the physiology of my study organisms. Therefore, I calculated the metabolism and the calcification rate of these three habitats. And my study organisms were two endemic seagrass species. On the top right, Posidonia oceanica, and Cymodocea nodosa on the bottom left, and in the middle, Halimeda incrassata, which is a tropical algae, which has become invasive in the Mediterranean. My study site was on the Balearic island, Mallorca, on the south coast, to be precise, with water depths of 2 to 6 meters. I chose this site because all my study organisms were encountered close by. However, my experiments were conducted in early spring, which resulted in a rather inactive state of Halimeda. Sorry. But it's here. Yeah. Well, sorry about that. So basically, for my field study, I set up 3 incubation bags, which consisted of hydrolabs and hobo sensors to control and report, document the environmental parameters, and 3 incubation bags in each habitat to take water samples. Okay. Well, water samples for total alkalinity and dissolved oxygen were taken at sunrise, at initially when setting up, at sunrise and at sunset. So normally that should be a video. However, there out of these 3 different samples, I calculated the metabolic rates, community respiration, gross primary production, and net community production, and the calcification rate within these habitats, which was done according to the alkalinity anomaly method, which basically has the assumption that total alkalinity is only affected by carbonate. The statistical analysis for my field study was done by a one-way ANOVA and Tukey HSD test. Coming to my results for the field study, I observed significant difference between the study organisms with the highest community respiration and the highest gross primary production per gram dry weight for Cymodocea nodosa, and the lowest for Halimeda, also due to its inactive state. The highest net community production I found in Posidonia oceanica, a negative community production, not net community production for Halimeda incrassata, and the results for Cymodocea nodosa fluctuated around 0. However, these results seem inflated due to the correction per gram biomass. That's why if we plot it per square meter, so per area of meadow, we can see that Posidonia actually, to my laboratory experiments, I chose a fully factorial design, which you can see in the right picture, so it's a bit messy. Therefore, I applied 3 different levels of temperature and CO2 to undertake short-term incubations in consecutive steps where oxygen and pH was recorded, and pre- and post-incubation samples of total alkalinity were taken. The statistical analysis were done with general linear mixed models with replicates as a random effect and ANOVA. My 3 steps were according to the ambient conditions I found in the field, which were 17 degrees and 400 ppm of CO2. Then, according to the RCP 4.5, an elevation of 3 degrees Celsius and 538 ppm, and for the high 8.5 RCP, an increase of 6 degrees and 936 ppm. Coming to the results, for the community respiration, I did not find a significant effect of the treatments alone or of the combination on the community respiration in my study organisms. However, visually or graphically, you can observe a trend, but it is not significant. In any case, Halimeda was the least active. The gross primary production was negatively affected by CO2, so by higher concentration of CO2 in Posidonia oceanica and Halimeda incrassata, which we can nicely see in the graph for Halimeda incrassata, which has this like step or staircase look. However, in each of my response variables, I observed an increase, in general increase in metabolic activity towards higher temperatures in Halimeda incrassata as it becomes more active. The net community production is affected mainly by progressing ocean acidification, so higher levels of CO2. In Cymodocea and Halimeda, I found a significant effect. However, I did not find a significant effect for Posidonia oceanica. But if we look at the 23 degrees graph for Posidonia, we see also these stepwise. However, in all my experiments, I found quite a big variance within the different plants. And we can, coming to the calcification rate, here I only show for Halimeda incrassata and leave out the other graphs. As Halimeda incrassata is calcifying and uses CO2, or to be more precise, carbonate out of the water column to build up its structure. However, unexpectedly, I did not find an effect of CO2 on the calcification rate in Halimeda, but I did find a temperature effect, which also correlates with the change from inactive to active state of the organism. I did find a significance for Posidonia oceanica and Cymodocea. However, this is mainly related to higher biomass and therefore more epiphytes sitting on the biomass, which contribute to this calcification rate. But the epiphytes are not as much as they contribute to it. However, bacteria in the sediment can also contribute to the calcification rate, and seagrasses can use carbonate out of the water column to use as a source of inorganic carbon. For my conclusion, I am confident with my experimental setup, the way I did it, as I did not find any effect of prolonged laboratory exposure. However, I had to do it due to logistic constraints, each incubation set up in the lab sequentially, so not simultaneously, which was mainly due to logistic constraints. The variance I talked about was quite high in between the replicates, which was also explained by my linear mixed models. In every case, the highest variance, or the variance was best explained with the model only including replicate as a random effect. The results and the results of metabolism of my metabolism and the calcification, which I presented here, are a proxy, or can be seen as a proxy for the overall idea of my project to as marine macrophytes as a carbon sink or carbon source. However, to draw a better conclusion to say, okay, they are a carbon sink or carbon source, we need to take into account that lacking data for total organic carbon, total inorganic carbon, and the sequestration rate into the sediment is lacking. However, seagrasses in general can be assumed to be carbon sinks, and my results go into this direction, as Posidonia oceanica has the highest potential to eliminate carbon out of the water column, mainly due to its higher biomass and the forming of big and dense meadows. However, the same we can say about Halimeda, however, only if it's in an active state, then it functions as a carbon sink, as it shows a high carbon turnover rate. But in its inactive state, it does not contribute to the carbon removal, but on the contrary, suffers from dissolution and can be therefore more seen as a carbon source, which is mainly happening in winter. However, in general, I can say that climate change and the effects of ocean acidification and elevated temperature in the ocean severely affects the organisms in my study. Coming to a prospect, suggestions towards my laboratory setup would be some more replicates, as I only had 3 replicates in my laboratory, longer adjustment time or adaptation time towards my laboratory setup, and the adjustment of mounting, because I observed shading due to my sensors in my laboratory. However, the temperatures applied in this study are still within the tolerance range of my organisms, but the predicted shift towards higher mean temperatures throughout the whole year can be assumed to alter the responses of organisms dramatically in summer, especially in summer, where they surpass the tolerance range of especially Posidonia oceanica. Furthermore, there is a need for investigation of synergistic effects and the effects on the scale of on the whole ecosystem. And next week I return to my institute and do the same field study again to compare July and the early spring. Furthermore, I'm expecting the results for total organic carbon and carbonate content in the plants, within the plants, and then I'm able to translate the measurements of total alkalinity to total inorganic carbon. And finally, I can calculate the sequestration rate within the plants. Thank you.